Okay, I know you're probably thinking, what in the hell is she going to be talking about today? Well, today I'm talking about some of the issues that come with using animals to analyze data, especially when the data analyzed is needed to develop strategies and treatments for human beings, for patients or potential patients. So, this new study may help to explain why so many encouraging results in animal studies don't hold up in human trials. This is a problem because it goes across the board. It affects policy. It affects how people are obviously treated in the hospital, what drugs they're treated with, how they're treated with those drugs, how frequently they're given the drugs. It also affects, it can also affect how the financial system works because a lot of these drugs that are put on the market, they are on the New York Stock Exchange. The companies have, they're public, and most of them haven't, what is it? They haven't hidden from being on the New York Stock Exchange. You know that they're there. They also are beholden to large corporations, pharmaceutical companies. So all of this comes to play when discussing whether or not these studies are reputable and if they are appropriate to be the background and the rationale for clinical trials. So the title of this is Study Questions Animal Data Underlining Many Clinical Trials. Okay. All right. Sorry about that. Many clinical trials. So this is coming from Science Mag again. I apologize for that. Okay. If you sign up to take part in a clinical trial, you can trust that scientists have solid evidence from animal studies that the test drug might work, right? After all, a study based on flimsy evidence would needlessly put your health at risk and potentially waste money. But your trust might be misguided, according to a new study. So-called investigator brochures, the documents that researchers produce to convince regulatory agencies and ethical review boards that a proposed trial is worth the risk, are often lacking crucial information about the efficacy of the proposed therapy in animal models. As a result, it's often impossible to tell how good the evidence is. This is incredibly alarming, says Shai Silderberg, Director of Research Quality at the National Institute of Neurological Disorders and Stroke in Bethesda, Maryland, who was not involved in the study. The work shows that decision makers for ethics related to clinical trials don't get the information they really need to evaluate those preclinical trials. Over the past few years, researchers have repeatedly shown that many animal studies lack scientific rigor. They are often prone to biases, for instance, and are sloppily reported in scientific journals. The team behind the new study looks specifically at the information researchers prepare to justify clinical trials in humans, which can cost millions of dollars. Investigator brochures are the main source of information for institutional review boards, or IRBs, the ethical panels that greenlight trials. So IRB is what certain institutions use to when you have a human impact, when you have humans involved in your study. IRBs are also included, along with many pages of other information, in drug trial applications with regulatory agencies such as the U.S. Food and Drug Administration in Silver Spring, Maryland. The brochures contain information about toxicology, pharmacology, and animal safety studies used to determine the potential risk a particular therapy may pose to humans. But they also include efficacy studies done in animal models to demonstrate a therapy's potential benefit. Investigator brochures aren't public documents, but a team led by Daniel Stretch of the Center for Ethics and Law in the Life Sciences at Hanover Medical School in Germany asked the chairs of 6 IRBs at German medical faculties, I guess they meant facilities, where they could see them, provided they signed confidentiality agreements and promised to report their findings in a way that would not identify individual trial sponsors, researchers, or candidate drugs. Yes, because that would be illegal to identify those people, all those individuals. 3 IRB chairpersons agreed to collaborate, giving the researchers access to 109 investigator brochures from trials approved between 2010 and 2016. Together, they included 708 efficacy studies in animals. The researchers read all of those studies, looking for things such as appropriate control groups and whether a sufficient number of animals had been used. They also checked whether the studies had been published in peer-reviewed journals. The team found that 89% of the animal studies were not published at all, making it impossible for the IRBs to know whether the study had been reviewed by other experts. Additionally, fewer than 5% included important information on whether bias-reducing methods such as randomization of the experimental groups were used. They report today in PLOS Biology. This could mean that the studies weren't set up to avoid bias, or that the measures were not reported in the investigator brochures, for instance, to keep the packets concise. Lastly, 82% of the brochures only reported studies that had positive effects. That suggests that trial sponsors leave out the less flattering studies, Stretch says. Even with promising drug candidates, there are often studies that show negative effects. For instance, if the dose was too low or the timing of the drug's administration wasn't ideal. Silderberg says that this finding is less surprising, however, because if the studies aren't positive, then you wouldn't go to an IRB to do a clinical trial. Yeah, so if you had 80%, or like if you do 10 studies and 8 of the 10 studies are showing negative results, you shouldn't go to IRB to, try to conduct a clinical trial. You should go back and try to obtain more positive results and increase your statistical significance of your study. Maybe increase the number of mice, maybe train other people in your lab to do the particular techniques so that you have a reproducibility of your study. If I'm just doing the same experiment 3, 4, 5 times, I'm getting similar results with very low error, then yeah, this experiment is repeatable because I've been repeating it myself. But if I do the experiment and I get results, and then you do the experiment and then you get the similar results, and we do it on different days, we do it at different times of the day, somebody else does the same experiment, they get similar results. Now this experiment is reproducible. Now you have statistical, confidence in what you're doing. You have confidence that the experiment is actually working. But they're saying here that a lot of people have negative data and they take the positive data, so they use that and they go to IRB and they get through. Stretch says he's surprised that IRBs and regulatory agencies accept the current situation. Then he says, why is nobody complaining about this? People are complaining about this, and they've been complaining about it for a very long time. They go on to another person, Spencer Hay, a bioethics researcher at Brigham and Women's Hospital and the Center for Bioethics at Harvard Medical School in Boston, agrees. If the brochures do not adequately describe the studies or do not provide adequate context that would allow the IRBs to make a sound judgment about their significance for human testing, then this is a serious problem, he said. Hay was not involved in the study, but he did complete postdoctoral training with 2 of the researchers on the paper. Wow. So there is bias, but he's still telling, the truth as a person working in his department. Although all the brochures came from German IRBs, the team believes the situation is likely to be similar throughout Europe and the United States. FDA declined to comment. Not surprised. The agency generally doesn't comment on specific studies, but evaluates them as part of the body of evidence to further our understanding about a particular issue and assist in our mission to protect public health, a spokesperson wrote in an email. It's not clear why investigator brochures often lack data, Stretch says. Some companies may prefer to keep information confidential because they worry about the competition, or they may believe that animal efficacy studies are too complex for most IRB members, which usually include non-scientists to review. IRBs may assume that no sponsor would invest in a clinical trial without convincing evidence and give the companies the benefit of the doubt rather than analyzing the animal studies thoroughly. But there could be reasons for companies to push trials without proper evidence, says Stretch, such as wanting to keep shareholders happy. I knew it was going to be some money coming in somewhere. I told you about these companies being on the New York Stock Exchange earlier. So here we go. Stretch says he plans to reach out to regulatory agencies, pharmaceutical companies, and clinical investigators to begin discussions about how to improve investigator brochures. If some studies cannot be peer-reviewed because they must be confidential, then at least IRB members should get better training to evaluate them, he says. The new study helps explain why so many results in animal studies don't hold up in human trials, says Malcolm McLeod, a neurologist at the University of Edinburgh. Less than 10% to 15% of clinical trials are successful. That's pretty sad, and I think there's a link to that. That's why it's highlighted in blue. The last thing they say is, in this article is improving the design, conduct, and reporting of preclinical research is of the utmost priority if we are to move ahead with all due haste in the development of novel treatments across a range of diseases. So they're talking about infectious diseases, genetic diseases, cancer, blood and bone-borne diseases, leukemia, sickle cell anemia, things like that. From all of that all the way up to flu and so forth. I hope this article wasn't too boring. It's just basically saying that there needs to be more careful observation and careful analysis and planning when doing the analysis of different diseases, for different genes that are changing, or you're manipulating different genes using CRISPR or using any other type of viral infection of the cells. Analyzing the results in animals, in mice and rats especially, is totally different than when you're using humans, because these mice are, for the most part, homogeneous, meaning that they're all pretty much identical genetically. They're clearly identical genetically, identical physiologically, and so forth. But in humans, you have a totally different, very diverse group that you're going to be using, unless you specify that you want to use a certain ethnic group, which you can't do in clinical trials due to discrimination. So you have to, I think what a lot of the scientists have to do is, like I was saying earlier, have data that's more reproducible in their preclinical research that they present. That way, the chances of them conducting a clinical trial and being successful during that trial are much higher. I think that's one way that they can do it. Anyway, you might have to read through this article again, maybe click on some of the links to understand more of what this article was talking about. If not, please, by all means, leave comments in the comment section. I would love to hear your feedback on this topic. Is science really going too far, or is there a way that there could be a solution to this issue? Just let me know in the comment section. Like always, like, comment, subscribe. If you're new to my channel, if this is your first time seeing my videos, welcome to my channel. This is Nicole Ali. I hope that you enjoyed this video. If you can, remember to subscribe. Please subscribe. Press the notification bell so you'll know when I post a new video. I'll talk to you all later. Have a great night.